This is an Edexcel GCSE past paper for Additional Science for Biology, B2 Topics 1 to 4, and it's for the higher tier. Question 1 is about using too much fertiliser and how it can cause environmental damage. We're asked to describe what effect the fertiliser has on tiny organisms called algae in rivers. Basically what happens is it causes an increase in the growth of algae. You could put an increase in the numbers of algae or an increase in the amount of algae. We're then asked to suggest why this effect causes some plants in rivers to die. You've got to consider we've got more algae growing, so basically less light gets through to the other plants and it stops them photosynthesizing. Part B is for 2 marks, so it's essential you put 2 relevant points in. Now when plants and algae die, they sink to the bottom of the rivers. We need to explain why this results in the removal of oxygen from the water. We need to consider that when any organism dies, it decays or decomposes. Now for decay and decomposition to occur, we need to involve microbes or microorganisms. Microbes and microorganisms are living things, therefore they respire. To respire, they're going to use the oxygen in the water. Finally, for part C, we're asked to name the effect that fertilisers have on the rivers. The word you're looking for there is eutrophication. Question 2 is a graph question. Edexcel like graphs, and it's quite feasible you could end up with 2 graphs on your paper. Now this graph is about the changes in the number of cases of skin cancer in males and females between 1975 and 2001. On the x-axis you've got the year, on the y-axis you've got your skin cancer cases per 100,000 of population, and you've got a plot for males and females. We're asked here for 2 marks to describe the changes in the pattern shown in the number of cases of skin cancer from 1975 to 2001. You need to discuss 2 aspects of the graph for your 2 marks. So, for the first mark we could say that the cases increase in both males and females, and they show a similar pattern. For the second mark we need to be more specific. Now we could talk about there being a decrease between 1987 and 1988. If you trace your finger up on the graph plots, you can actually see that both females and males dip slightly. And then from 1988 to 2001 there is a clear increase in the skin cancer cases. Part B talks about skin cancer being a living indicator of the impact of human activity on the environment. We're asked to explain what is meant by a living indicator, again for 2 marks. A living indicator is a living organism whose distribution is related to a change in the environment. 1 mark will get you the fact about it being a living organism. The second bit is about the distribution being related to the change in the environment. Question 3. Here we've got a picture of a guy called Leonard Hayflick who researched human cell division. And one part of his research is now called the Hayflick limit that you'll have met in lessons. We're asked to describe what happens to a normal human cell when it reaches the Hayflick limit. All you need to put is it stops dividing. Now cancer cells and also stem cells do not have a Hayflick limit. We're asked to explain why this makes cancer cells so dangerous. 2 marks, 2 relevant points. First of all, they don't stop dividing. There's 1 mark. What's the downside of this? Well, it leads to an uncontrolled growth and can end up as a tumour. Part 2. Here we look at the stem cell side of things. Stem cells can be taken from embryos that are a few days old. We're asked to suggest 2 reasons why some scientists think that these cells could be very useful to humans. For the first one, they have no Hayflick limit, so they carry on dividing. Secondly, they can differentiate or grow into any sort of cell. So your stem cell is not specialised, but it can differentiate into any sort of cell you want. This means they can be used to replace damaged cells, tissues or organs. Finally, we're asked to suggest a reason why some people are worried about using embryos for stem cell research. Remember, stem cells come from an embryo. and to get them it results in the death of the embryo, which is unethical. Question 4, graph number 2. A farmer thinks he can increase the yield of tomatoes by increasing the light intensity in his greenhouses. The graph shows the effect of increasing light intensity on the rate of photosynthesis. Light intensity is on the x-axis, rate of photosynthesis on the y. Using the information from the graph, you've got to describe the effect of increasing light intensity on the rate of photosynthesis. 2 marks. So, we start off by saying when the light intensity increases, it increases the rate of photosynthesis, as indicated on the graph. However, the graph levels off. So, we then say until the light intensity is approximately 67 units. If you look where the flat bit starts on the graph and you use a ruler, this will link in with about 67, 68 units. So that's when the rate levels off, which shows that light is a limiting factor. Part B. An advisor suggested to the farmer that another limiting factor could be preventing further increase in photosynthesis. We're asked to name the limiting factor and describe its effect. Here I've put several limiting factors down. Firstly, temperature. Temperature could be the limiting factor, and it can be maintained by either heating or cooling the greenhouse. Carbon dioxide. Plants need carbon dioxide to photosynthesize. Now here we need to increase the CO2. We could do it by combustion, by burning something in the greenhouse, or perhaps a manure heap. Here the decomposers that live in the manure heap, decomposing it, will respire and hence increase the CO2. The soil might be lacking in minerals, so we could add some fertiliser. Or water. Plants need water for photosynthesis. We can simply water the plants. Question 5 is all about fermenters, and you've got a rather complicated looking diagram. Now penicillin is an antibiotic that is needed in large quantities. To make penicillin, the fungus, Penicillium, is grown in a fermenter. The antibiotic can then be extracted, and we've got this diagram of our fermenter. First of all, we're asked to consider how the temperature in the fermenter is kept at a constant 30 degrees Celsius. If you look, you can see a cold water jacket. The point of that is to keep the temperature constant at 30 degrees Celsius, so we simply write the water jacket keeps it cool. We then need to explain why the pH inside the fermenter is kept at 6.5. Basically, it's the optimum pH for enzyme activity. That'll get you a mark. The second mark, you need to link that in with the relevance. Well, then the right pH for enzyme activity will then help in the growth of microorganisms. We then look at the conditions inside the fermenter and the fact they must be aseptic. We have to state what is meant by aseptic conditions, again 2 marks. Firstly, we need to say there are no unwanted microorganisms present. What will that mean? Well, it will mean there's no contamination. The materials entering the fermenter are sterile. Now, how are these aseptic conditions maintained? Again for 2 marks. First of all, the fermenter is steam clean between batches. It's also sealed, the air is filtered, and the food is sterilised. Finally, question 6. This is for 4 marks, and we're told that athletes get cramp during exercise. We need to explain why the body needs extra oxygen to recover from cramp. 4 marks, you need to write at least 4 pieces of relevant information. First of all, we can start by defining what cramp is. Well, it's caused by lactic acid. Where does the lactic acid come from? It comes from anaerobic respiration. We then need to look at why the extra oxygen is necessary. Well, oxygen is needed to break down the lactic acid into carbon dioxide and water. This is known as oxygen debt. So, first mark for the bit about cramp, second mark for the oxygen breaking down the lactic acid into CO2 and water, third mark, it's known as oxygen debt, and finally, where does it all happen? The lactic acid is broken down in the liver. That will give you